Maria Francesca Abbate. She's a computational immunologist with a statistical physics background, specializing in B-cell receptor repertoire analysis and antibody discovery. During her PhD, which was in collaboration with Sanofi, she developed a computational approach to identify antigen-specific BCRs. And this work contributed to a complete antibody discovery workflow, leading to the identification of potential therapeutic antibody candidates. And she's going to tell us all about it right now. Welcome.
Thank you. So, great talk, Stephen. So I will share the screen. Thank you for giving me the organizer. What? Sorry, I think the connection not great. Okay. Today I'm gonna talk about the work I did during my PhD that was done at the École Normale Supérieure in Paris under the supervision of Alexandra Volchak and Thierry Morat. And it was done in collaboration with Sanofi. And in particular, during this talk, I will present in the first part what is written in the first part of this title. And so I will show you a computational pipeline that we developed to detect the antigen-specific B-cell receptor sequences starting from one repertoire. And then the second part of the talk will be this antibody discovery. And so I will show you how to integrate this computational pipeline into an entire antibody discovery workflow. So I will really briefly just give a little bit of introduction. So we know that the B cells, as the T cells, are the cell of the adaptive immune system. And so in their DNA, they have a specific region that is the one that codes for the receptor that they express on the surface, that it's unique. And this diversity is reached through a process that is called the VDG recombination that happens in the bone marrow. And thanks to this process, different gene segments are connected to each other. And like this, we end up with a huge variability of different receptors that we can have. So the B-cell receptor contains two chains, the heavy chain and the light chain, and they rearrange together in order to form the structure of the receptor that is the same structure of an antibody. And the VDJ region is really the one that gives the specificity to the antibody to detect many different pathogens. So this diversity that is reached thanks to the VDJ recombination, it is not enough to detect all the possible antigen that we can encounter in our lifespan. And so B-cell needs another layer of diversification that is reached through the affinity maturation. So what happens is that when an antigen invades a host, the B cell goes to the lymph nodes towards a region that is called germinal center, and they start to proliferate and mutate, and then they compete for the help of a T cell helper that will give the signal to the B cell to die for apoptosis if they acquire the deleterious mutations, or to re-enter and do another cycle of affinity maturation, or to release as memory of plasma cell if they are good and ready to fight the infection. And so like this, we end up with a pool of different B cells that can be grouped in lineages that can be represented in a tree in which the root is the ancestor, so the naive B cells, and all the nodes are the mutated B cells. So one key question here in biology can be given this pool of all these different B cell and each B cell receptor with its unique specificity, how to identify the specific receptor or specific antibodies that react against a given target. So in order to do this, we need to collect some data. We can start from a blood sample, for example, and then thanks to the PCR amplification of the DNA or the RNA and to the sequencing machine, we can end up with the data that look like this. So the B-cell receptor repertoire, there are a list of different sequences. So in my PhD, my objective was to detect the sequences that are antigen-specific, starting from this repertoire, and in particular doing this using only one time point, that is the peak of the infection. So in order to develop this pipeline, we used some public data set of 5 humans that were vaccinated against the flu vaccine of 2008. And in particular, we had longitudinal data, so before and after the infection. And we used only the CDR3 of the heavy chain. The pipeline works only for the CDR3 of the heavy chain. So in order to develop this pipeline, we had to focus our attention on some features, and we started from nearest neighbors. So what is our definition of a neighbor? We can take two amino acid CDR3 sequences, and we can call them neighbors if they have one amino acid mismatch. And so this can be represented in a graph as two nodes connected by one link. But then we will add some multiplicity, that is the number of distinct nucleotide sequences that are in the data set that translate for that amino acid sequence. So in this example, the sequence on the top will end up with 4 neighbors rather than just one. Then why we decided to focus on neighbors? This is for a biological reason. So we saw that at the cellular level, what happens is the affinity maturation. So the cells expand and mutate, resulting in lineages. And this can be translated at the sequence level with a highly connected graph of similar sequences. And so the idea is, let's try to find these expanded graph being agnostic on the reason that a big lineage that was expanding or from different but similar lineages that were expanded towards a similar direction. So now we can look at some comparison of the features, and here in this plot you can see the distribution of frequency for the 5 patients in the background, while the thick line are the average between the different patients at day 0 versus day 7. And what you can notice here is that the distribution of frequency doesn't really change over time. I mean, it changes, but really a little. Distribution for a number of neighbors, we can see here that there is a clear shift between which is considered the peak of the infection. And so it was kind of natural for us to put a threshold somewhere here and look into the sequences that are above the threshold to check whether there were some potential antigen-specific sequences. Now there is a problem, because in order to put this threshold, I need day 0. And at the beginning, I told you that I wanted to build a pipeline that was able to work in using only one time point. And if using only the peak of the infection, we need to parameterize a null model. So the null model that we used is a generative model that was produced by former people in our group. And what it does is that it computes the probability of occurrence of any given sequence. And so having this probability, it is straightforward to compute the expected number of neighbors of any given sequence. And with expected, what I mean is that if I have a sequence has star, how many neighbors I would expect this sequence would have in a healthy patient, so in absence of immunization. And this is simply the sum of the probability of occurrence of all the sequences that belong to the ensemble of neighboring of my sequence as star. And of course, multiplied by the total number of the sequences that I have in my data set. And now we can look at the null model, the same distribution that we saw before, so the distribution of number of neighbors, but computed this time, the null model versus day 0. And we can see that they agree pretty well. And so in this case, we don't need to use day 0 anymore, in the sense that we don't need longitudinal data that are provided from the same lab anymore. Now, just to recapitulate how the pipeline works, that is called STAR, and available on GitHub. That is single time point antibody recognition. It starts computing the number of neighbors of each sequence using the multiplicity in nucleotide, then infer the threshold using the generative model. And then we added an additional step here. So for the sequences that are above the threshold, we infer the clusters of similar sequences using Levenshtein distance. And in particular here, we exclude all the clusters that have less than 10 sequences. And you can see here an example of an actual output of the first patient. We can look now at some results. And here in this plot, you have the number of hits, so the number of sequences that are output of the pipeline when we apply the pipeline to all the patients and to all the data that we had. So first of all, we can see that there is nice peak at day 7. That is the peak of the immune response, and that there are no hits before the vaccine. So this was really good. And then Now, if we focus on the first patient and we want to see how the sequences output of the first patient are arranged in space, they look like this. So this cluster of highly connected sequences, and you can see the logo sequence associated to the cluster on the top. And in particular, if we focus on the 4 nodes that are circled in blue, these are 4 amino acid CDR3 sequences that belong to 4 antibodies that were tested experimentally by another lab, the same lab that produced this data set in the first place. And in particular, these 4 antibodies are the one with the highest affinity against the flu vaccine among the one that they decided to test. So this was great for us because, first of all, we had proof that the pipeline was working. And then also we had some ground truth. We had some data set for which we knew that there was some binding and also that the affinity was pretty high. And so we said, okay, let's come back to the data and let's see if we can find some interesting features. And in particular, what you can see here, these are the same sequences of the cluster before, but by gene usage. So it is really interesting to see that very similar sequence, and in some cases even exactly the same sequence in amino acid, is shared by many different genes. And this means that they cannot belong to the same lineages. And so we try to infer the lineages, and we reconstruct the lineage tree associated to this cluster of sequence. And here in this plot, the color code is the same of the plot of the above. So same color, it means same amino acid CDR3, and the different nodes instead are different nucleotide sequence. And the size is simply proportional to the number of reads that was in the data set. Here instead, if we focus only on one amino acid sequence, that is the most represented one, this pink, we traced the trajectory of this.
Там, короче, ты...
Okay. We can trace the trajectory of this sequence, how it expanded in time. And in particular here, each line is a different nucleotide sequence, but all of them translate into the same amino acid CDR3. And in particular, the different color are associated with different V genes. So we can really see that they independently expanded. And this is really a hint of some convergent selection that was going on within this patient. So now we can move to the second part of the talk, so which I will show how to integrate the pipeline into the antibody discovery workflow. And in particular, we started from the immunization that was done internally in Sanofi to the repertoire. Then we applied STAR. And then we came back to the wet lab in which we tested the sequences that were output of the pipeline STAR. We tested for their affinity against the target that we decided with which we immunized the mice. So why it is important to have monoclonal antibodies? So they are highly specific to a single antigen, this we know, but it was really see that they can be key in cancer, but also in some autoimmune disease, as well as some chronic conditions. And as of 2025, there are over 100 different monoclonal antibodies that are approved for therapeutics. The immunization workflow that we used in Sanofi is represented here. So we had 5 triani mice, so humanized mice that we injected with 4 shots of injection of the target GFRAL, that is a receptor. And then we collected blood sample every time 4 days before and after each injection. And of this blood sample, we performed the bulk sequences every time of the heavy and the light chain. Then for some specific days, we also had the paired sequences to match the heavy with the light chain. And then at day 67, we also collected some splenocytes on which we performed the bulk, the paired, and an additional sorting that was done for antigen positive and antigen negative kidneys. So why did we choose this target, GFRAL? This is a receptor that was discovered recently to be the receptor associated with a cytokine that is called the GDF15. This cytokine is known to be associated with the regulation of the appetite. So in general, everything that relates the sensation of nausea, the food intake, and so consequently also the body weight loss or receptor gain. And so the idea of monoclonal antibodies against GFRAL could work in two different ways, because we can think about agonist monoclonal antibodies against GFRAL, and so that they activate GFRAL, and this can be useful for all the patients that suffer from conditions like diabetes or obesity in order to lose weight, but also the opposite is true. So if we have antagonist monoclonal antibodies against GFRAL that suppress the activity of GFRAL, this can be useful for all the patients that suffer from conditions like cancer cachexia or anorexia. So we can look now at some results, and in particular, we had the room to test over 200 monoclonal antibodies against GFRAL, and we choose them according to three different criteria. Of course, the first one are the sequences that were output of star, and this resulted in a 50% of binders. Then the second criteria is what we call the bulk same cluster. Basically, we took some CDR3 of the heavy chain that were very similar of the sequence that were output of star, but they were not present in the paired dataset, and so we matched these sequences with the same light of the corresponding output of star. And so this resulted in a 20% of binding, which is really interesting if we think that none of these sequences was actually present in the single cell dataset. So we had to match the heavy and the light chain. And then the third criteria is completely independent from the bulk data. And it's just the single cell, coming from the single cell sequences, we just took the most frequent, and these resulted in 80% of binders. If we want to see from where the response is coming from, here we can see where the binders were coming from. And in particular, the highest peak is from the spleen, and this is not surprising. But the second peak is this gray bar. And so these are the sequences that were not present in the single cell dataset. So again, this was really interesting for us. If we want to look now at some features of the binders, we checked whether there was some correlation between the single cell counts and the affinity. And the affinity here is represented with KD. And the lower the KD, the higher is the affinity against the target. And we saw that there was absolutely no correlation between the two features, while instead we observed some sharing between the binders. And in fact, here there are 13 different binders that are shared by at least two different mice. But in some cases, we also have the three mice together that express the same binders. Then, since we had this data set, this list of monoclonal antibodies with all the KD, we decided to investigate the software AlphaFold 3 and check whether it was capable of distinguish between the binders of the non-binders, using as input the antibody, so the heavy and the light chain, but also the antigen that of course we knew. And so the result is the following, that in most cases, AlphaFold was not able to detect, to distinguish between binders and non-binders. And in particular, this we can see it here with this score of AlphaFold that is called IPTM, that we can use it as a proxy for the affinity predicted by AlphaFold. So in most cases, the score is really, really low, but in the cases in which the score is high, so according to AlphaFold, the complex was, the antibody was a binder, then it was really a binder, and it was also having a very low KD. In fact, we have a correlation that is not pretty high, but it's still of minus 0.5. So the take-home message from this for me is that there are many false negatives, but there are only a few false positives. So it can be used, the software, as an additional screening in an antibody discovery workflow. So here I just wanted to show you two examples of the structures that are predicted by AlphaFold, but are depicted here with the software Pymol. And you can see at the top the antigen, while the antibody is at the bottom. And like this, I would like to conclude. So just to summarize, I presented you a computational pipeline to detect the antigen-specific sequences starting from one repertoire of one time point of bulk data. that was experimentally validated with good performances. And then additionally, we found some internal convergent selection within one subject, but also external, so across the different mice. And we end up with a list of monoclonal antibodies that are specific against GFRAL that can be potentially tested by other lab or pharmaceutical company and potentially used for therapeutics. And I just want to finish saying that in general, the computational approach and using computational pipeline inside an antibody discovery process can really have an impact on the timeline and on the efficiency of this process. Thank you. I will be happy to take any questions.
Yes, thank you so much. Are there immediate questions from the audience? Maria, could you go again once more over the features that go into the STAR algorithm? Like, based on what assumptions are you identifying antigen-specific clones?
Yeah, it's like, okay, maybe it's shown here. So
Yeah.
the idea is that we compute the number of neighbors. So actually it works in two ways, because STAR, there is like a fast STAR. The one that I presented is fast STAR.
Okay.
That is more restrictive, and then there is full STAR that is less restrictive and more solid from the point of view of the statistics. So the idea is that for fast STAR, that is the one that is really quick and can run in 1 hour. We just compute the number of neighbors according to the definition that I said, and then within the sequences that are above the threshold, and here the threshold can change from patient to patient, but we saw some consistency across the different patients, and the threshold can always be inferred from the null model, but I mean can be also inferred from some baseline, just some data set of some healthy patient provided by different labs. I mean, this it doesn't matter so much, because then the idea is that above the threshold we do again the clustering of levest and distance, and I mean just we kind of fine-tune different parameters to choose what was the optimal distance to use, like the levest, why the levest and distance and not the Hamming distance. But I mean, generally the idea is this. Then we infer the cluster, and we take only the cluster that have a minimum of size 10 that are all above the threshold. So not only my sequence needs to have a lot of neighbors, but also the cluster needs to be really dense, because the risk sometimes is that if there are, I don't know, some PCR amplification errors, I mean, that there are always, I mean, this is impossible to remove, but the risk is that maybe one sequence has a lot of neighbors, but then the cluster is kind of linear. So it's not really interesting. I don't know if I was clear.
Okay, so the biological intuition of why this works is then because of the German center reaction. We have clonal expansion, and you're trying to find the most likely clusters that are related to the antigen.
The idea is just that the big cluster, it's reasonable that they are involved in the immunization, and the problem is really like how to define big, because of course you can have, I mean, it's not really important big, but the important is expanded, and so for this expanded you usually need the timeline. And so in order to find expanded but without timeline, then you need the null model. And the other pipeline that is the full STAR actually does a bit more than the thing that I explained. So actually for each sequence it computes a p-value of how unexpected is that number of neighbors. So this is the idea.
Okay.
But do you, I mean, what did you do to verify that you are accurately inferring day 0 when you don't have it?
No, we had it. So the idea is that we had the zero, and we saw that the null model was able to simulate the zero across different patients.
But
And
yeah,
so,
but I'm asking based on what? I didn't quite understand how you determined that it was able to.
just because it was able to reproduce, sorry, it's here. It was able to reproduce the distribution in terms of number of neighbors on average.
So but that's just by count of neighbors, not by their representation of what sequences are included.
Exactly.
Is that,
Exactly. It's just,
I mean, is that
it's just
sufficient to say that it's doing a good job at accurately representing that patient's day zero?
no. In fact, I mean, we don't use it as a generative model to generate repertoire, because the problem is that this model also takes an assumption that the sequences are generated independently from each other, which is not the case for B cell because they form lineages. So, of course, I don't think that this simulates a repertoire of a patient, but for the purpose of number of neighbors and the pipeline to work in, that at the end I just need a number in where to put the threshold, and then I will infer again. I mean, I don't need a number that needs to be exactly the real number, because then I will do more analysis on top of that. And so the idea is that I don't have something that really simulates a repertoire, but I have something that is good enough for my purpose.
But I mean, I guess I can buy that, but then you're inferring that the thing that you're finding in your day 7 is actually an expansion. But that's an inference based on an inference, right? Because you don't know anything about that actual cluster within day 0.
I
Like
mean,
you don't know that cluster wasn't actually there, right? Before
no, I mean,
getting-
this we did it. Of course, we did it because we had the day, and then we use it also to fine tune the parameter that we choose. But the real test was done after, when we use a completely different data set. That was the one that was developed in Sanofi, and in that case, we like the one that we tested experimentally ourselves. I didn't use any information. Like there, I just took the repertoire day by day, completely independently coming before, and I just applied the pipeline with the parameters as they were inferred from this data set.
Okay, thanks.
Anastas?
Hello, great talk. I'm really excited about these results, but I was interested just technically. You picked Löwenstein distance, and it's often used. Did you study different editing distances? Have you any
Yes.
criterion for comparison between the different editing distances?
Yes. So first of all, we tried with different distances. So the interesting thing of the Löwenstein distance, it was again connected to the convergence selection, because what we saw is that in some cases there were some using only Hamming distance, for example, there were some cluster above the threshold that were not connected to each other because they were cluster of sequences with a fixed length that was different. One was 18 and one was 19. But then when we looked into the cluster and into the logo sequence of the different cluster, they were identical, but for one amino acid less or more. And so the idea was that it was including the knowledge of the convergent selection, because I mean, it's quite rare that just for mutation you can end up with 2 amino acid less. The idea it was again that acknowledging the convergent selection, it was better to use the Löwenstein distance, and we didn't use it from the beginning just because for the entire bulk data set it was taking too long using the Löwenstein distance. And then we inferred the optimal threshold. for clustering the sequences, and this depends on the length of the sequence of the amino acid CDR3, but is around 1 and 2 if if the sequence is very long. Sorry. And then we also tried different, like we tried to compare different way of distancing. And what I mean with this is that we kind of applied the pipeline star to the lineages or to the cluster. And so we defined an average distance and try to find neighboring instead of sequence, but we didn't have any results. So at the end, we just stick with this one now.
Thank you.
Great. Do we have more questions?
I reply.
Maybe on one last question, on one of the slides you said 22 of the binders were not present in the single cell data. What did you mean by that?
Yeah.
Didn't you have to yeah. So where did they come
Yeah,
from?
so
They were not in the single cell data.
yeah, they came from so the heavy chain came from the bulk.
Sure.
And I mean the CDR3 of the heavy chain was very similar to the CDR3. It's here. So the CDR3 of the heavy chain was very similar to the CDR3 that was output of the pipeline star.
Yeah.
And most of the sequences that were output of the pipeline star were also present in the single cell dataset. So this one that I tested, the one that I called the bulk chain cluster, the heavy chain was from the bulk, and they were not present in the single cell data. So I used to match the light. I just used the most the light of the most similar heavy chain that was present in the single cell data.
And that worked, but that's surprising. That's nice. Yeah.
Yeah, in fact, I mean, we were quite surprised. But yes, because they were, I mean, they were not randomly chosen. They were still sequences that were very similar to the one that were in output.
Yeah.
So we had confidence that maybe the heavy was the correct one, and then we just used the light that we had.
Yeah.
So like this, we matched this, and it was pretty nice. And actually here you can see that if you see the KD, that these gray points that are still not present in the single cell dataset, they can reach very low value of KD.
Yeah.
So
Yeah. No, it's nice. Nice. Jan, as the last question.
Is there a question from?
Hi, thank you. That was a very interesting talk. You mentioned that your method was only working for very dense clusters, right? How do you quantify this density? Do you just take like the number of present sequences, considering all possible ones, or?
So this really depends on the data set, and the quantification, it really also depends on the threshold. So the threshold is really consistent across the different patients, and is always around, in terms of normalized number of neighbors, it is always around 10 to the minus 2. And so the problem is that using pipeline, that is the one that represents the data, is not deep enough, you have 0 hits. So they really need to be very dense. But then this is why we also went to full star, this other pipeline that instead it doesn't cluster the sequence. It just compute the number of neighbors of each sequence and then compute the expected number of neighbors of each sequence using the generative model, and then it computes the p-value. And then in this way, I don't have to cluster anymore. I just rank the sequence in terms of p-value, and I take the one with some false discovery rate. And so in this case, I just take this can work only also in data sets that are not deep enough, but it's less precise.
So you don't explicitly have like a density number that you use in your calculations somewhere, or?
Yes, it's in terms of normalized, it's 10 to the minus 2. Of course, normalized I mean just like the cluster, the number of the cluster divided by the total number of the sequence that you have in the dataset.
Okay, I see. Thank you. Thank you.
Thank you for all the questions, and thanks again to the two speakers today. Very exciting talks. We are coming to the end of this seminar series, and we will be back in a month. Thanks, everyone. Thank you, Maria. Thank you, Steve. Thank you.
Thank you so much.
Thanks to your audience. Bye-bye.
Bye.
Thank you.